Hi everyone, my name is Matt. I'm a postdoctoral scientist at the University of Oxford, and I've spent my career as a scientist working on fighting antibiotic resistance with both chemistry and biology. Hopefully, today I'll tell you a bit about my journey through chemistry, as well as give you an idea of the science of antibiotic resistance, and try to explain what we're doing to combat it through both natural and man made solutions as scientists. So, starting off with my journey, I started off with science at school, as many of you did. I did my GCSEs and then A levels at state schools near Stoke-on-Trent. I was always into science, and it was a big part of my life. I was part of my school's science and engineering club. I chose to do the full suite of maths, physics, biology, and of course chemistry for my choices at A level. And I did this knowing it would set me up to continue on with science at university in whatever capacity that I wanted to do. And when I finished and I was deciding to go to university, I had a lot of options for what course I wanted to do. I thought about going to be a medical doctor, but I think I was a little bit too squeamish for that. And I had thought about biology related degrees as I enjoyed it as a subject and I enjoyed thinking about the human body and how we could treat it. But eventually, I went to the University of Warwick to study chemistry. I chose chemistry as it was challenging, but I still saw some of the real world applications of it. I was especially interested in learning how to make molecules that could be used as medicines that could help people. And in that way, I guess I was trying to be a doctor without being a doctor. I did an MChem degree, which is essentially 3 years of taught course material, followed by a fourth year research master's degree, with the two combined into one course. Um, in years 1 and 2 of the degree, I got a general chemistry education focused on the three main areas: organic chemistry, inorganic chemistry, and physical chemistry. Eventually, though, I ended up specializing in what we call organic synthesis, where I learned how to make molecules using lots of different chemical reactions, and also medicinal chemistry, which focuses on understanding how to make molecules that are really good medicines. In my third year, I was very fortunate to do a full research placement at a full year research placement at a big pharmaceutical company called GlaxoSmithKline or GSK in Stevenage, just outside London. This was a great experience, and I got to learn how to do chemical synthesis in a hands-on environment, which gave me lots of experience of organic chemistry that maybe I didn't get at university. I also got to use analytical techniques you may know, such as mass spectrometry, chromatography, and NMR spectroscopy. And I got to use these equipment well before I would have got to use them in my university course. And of course, there's no better place to be trained to make medicinal molecules than at a big pharmaceutical company. They had so much equipment and so many expertise that I could draw on that I got a really great education in that sort of discipline. Following that, I returned to Warwick for my master's research year, where I did a placement in the lab of Professor Greg Chalice, who works in natural products. And during this project, I did more chemical synthesis. But then, when I'd finished the chemical synthesis, I took the molecules that I had made and tested them against bacteria. And this really piqued my interest. For a long time, I'd wanted to be a synthetic medicinal chemist who worked exclusively on organic molecules. But actually, the process of testing these molecules on bacteria was really fun, and I really enjoyed learning about the biology of the bacteria themselves. With that in mind, I decided to apply to do a PhD in the same lab, and I was lucky to get funded by a doctoral training program called MIBTP. A doctoral training program is the most common way to get funded to do a PhD in the UK these days. It usually involves 3 years of research with a year's worth of training in between them, with most people doing that year of training at the start. So, for example, I got to do lots of different courses where I learnt about biology and learnt skills outside of chemistry. And I also got to learn some more real world skills. So, I did a placement at the business development arm of the university, Warwick Ventures, where I learnt a lot about commercializing research and how businesses see science. And it was a really valuable experience for me that I draw upon still as I work as a scientist. So during my PhD, I did nearly 4 years of research in the field of natural products or natural molecules for combating antibiotic resistance. And as I did that, my research gradually began to change. I started off doing chemical synthesis as I had before, because this is what I knew and what I was good at. But then again, we wanted to test those molecules once I'd made them, and we tested them on some enzymes from bacteria. And I really enjoyed working with these enzymes. And I started to really enjoy understanding and exploiting the enzymes that I was working with. And this project really, by the end of it, I'd turned from a synthetic organic chemist into what was nearly a fully fledged biochemist. And I really enjoyed it, and I wanted to learn more and more and do more of the biochemistry. And that led me to where I am today. So after finishing my PhD, I was able to get a job as a researcher here at the University of Oxford in the lab of Professor Chris Schofield as part of the newly formed INEOS Oxford Institute for Antimicrobial Research, where I remain to be a biochemist. I do a lot of work with enzymes, trying to understand them, characterize their reactions, and hoping to find molecules that can inhibit them to prevent antibiotic resistance and become the next generation of antibiotic therapies. So that was a brief summary of my journey as a chemist through to being a biochemist in an academic environment. And this is kind of the typical structure of a career in academia. So going through your undergraduate degree, then getting your PhD, and then doing postdoctoral research, with the next step being working towards being a lecturer or group leader, and then eventually a full professor. But that's not the only option, and there are many routes to take following any of these steps. A chemistry degree can set you up for so many different jobs, and it gives you important problem solving, mathematics, and critical thinking skills that lots of different industries are looking for. And these can be in science, doing chemistry, in more conventional environments, so things like pharmaceutical or agrochemical companies, amongst others. And I know plenty of people from my degree and PhD who've gone this route. There are what we call science adjacent careers as well, so things where scientific knowledge is important, and scientific knowledge is really in demand in things like journalism, scientific publishing, governmental jobs, as well as things like scientific patent law and business development, which I got to experience at Warwick Ventures, and I know quite a few people who've taken this route from their degree. And finally, you can go outside of science altogether. This is especially common for sort of mathematics-based jobs, things like finance, as the skills that you've learned doing chemistry tend to lend themselves really well to these sorts of jobs. So hopefully that's giving you an idea of what my journey through chemistry has been, and hopefully what your journey could potentially look like. So let's start to move on to talking about science, particularly some of my research in fighting antibiotic resistance with respect to natural and man-made solutions. So let's first set the scene by talking about what antibiotic resistance actually is, why it's such a problem, and why we want to combat it. So bacteria have been around since before humans, and we've coexisted with them for thousands and thousands of years. Most bacteria are a perfectly normal and healthy part of the world. You'll find them everywhere, from rivers to soil to even in your own body. And these so-called good bacteria are really vital for the survival of lots of different ecosystems. Unfortunately, there have been bacteria across the years that are really harmful to human health and can cause lots of different side effects, including death. And there have been massive plagues throughout history caused by bacteria that you may have heard of. So the Black Plague, bubonic plague, and even to this day, you still find outbreaks of diseases caused by bacteria. So things like cholera, you may have heard of. And even recently, you may have heard on the news about strep A. So this is a bacteria which has been infecting and harming lots of young children across the United Kingdom, and showing that bacterial infections are still a really big problem for human health. Luckily, scientists have developed medicines to fend off bacteria, called antibiotics. You've probably heard of antibiotics and may have even taken some. Humans discovered lots of antibiotics in the 1900s, and it was kind of thought to be the end for bacterial infections. However, when you treat bacteria with antibiotics, it introduces a selection pressure for the bacteria, which allows them to evolve to be antibiotic resistant. And this evolution here is represented by the different colours of bacteria on the slide. And as you use more and more antibiotics over time, these resistant strains tend to become the dominant population of bacteria as you slowly kill off the non-resistant ones. And this means that the antibiotic resistant bacteria become really prevalent and that your antibiotics work less and less well over time. And this has become a precarious position nowadays, with many bacteria now resistant to more than one type of antibiotic. So you might have heard of MRSA, and that stands for multi-drug resistant Staphylococcus aureus. So this is just a type of bacteria that's got lots and lots of different antibiotic resistance to different antibiotic classes. And the deaths worldwide due to antibiotic resistant infections are expected to rise to nearly 10 million a year by the year 2050. So this is becoming a really, really big issue. And this problem has been massively accelerated by our use of antibiotics across the world. So, for instance, we massively overuse antibiotics in humans and animals. The use in animals might surprise you a little bit, but around 80% of all antibiotics by mass are used in animals. And a lot of them are used not to cure illnesses in the animals, but actually as a preventative measure to stop the animals getting infected in the first place. But because you're using antibiotics on healthy animals, this actually accelerates the growth of resistant bacteria without actually providing any positive benefit. And bacteria that infect animals can actually then infect humans down the line. So it's not just a problem for animals, that's a big problem for humans. Another problem is not using antibiotics as prescribed by the doctor or as instructed by pharmaceutical companies who've tested them. You should always try to finish the full course of antibiotics and then only take them when necessary, as described by your doctor. If you use antibiotics to try and cure something like a virus, an infection caused by a virus like the flu or COVID-19, not only will it not cure your illness, but you might actually be contributing to creating antibiotic-resistant bacteria. The last one is poor hygiene. This is particularly in hospital environments, so having bad hygiene allows the bacteria to transmit between different people, which means you need to use more antibiotics. And also, if there are resistant strains in one person, it means it can transmit to another person and then proliferate really quickly. So why can't we solve the problem? Why can't we just make new antibiotics, for instance? Well, many antibiotics were discovered in the 1900s, but since the 1990s, there's been a huge slowdown in finding new antibiotics. This is partly due to it being harder and harder to find new molecules that act as antibiotics, because also the easy molecules and the low-hanging fruits have already been picked. But the other problem is there isn't big money in antibiotics. And to do this, you have to kind of understand the concept of last resort antibiotics. So when we make new antibiotics, they are often designated as last resort. And this means we don't want to use them on bacteria unless it's a really serious case where they are resistant to lots of others antibiotics. And this is because if we start using a new antibiotic, that resistance is going to develop, and then it's just compounding the problem going forward. And big pharmaceutical companies don't want to invest millions or billions of pounds trying to create an antibiotic that then we don't want to use because it'll sit on the shelf and won't get sold. So there isn't really an economic incentive for companies to discover new antibiotics at the moment. Most recently, it's been up to academic environments, so places like universities, to do the research into new antibiotics. So let's take you through two types of solutions that academics have been working on and that I've worked on throughout my career. and these are natural solutions and man-made solutions. Let's start with the natural solutions then, and these typically refer to a class of molecules called natural products. So natural products are these extraordinarily complex molecules that you can see on the slide. They have lots of different atoms connected in various different ways, and they have this extraordinarily complex structures that are very hard to replicate by humans. The molecule on the left you might recognize. That's because it's a variant of penicillin. And penicillin's the structure of which was actually discovered here at the University of Oxford, and gives us a big historical appreciation for antibiotics here. So natural products are produced by a wide variety of organisms, including plants and fungus. But most commonly, we find antibiotics from other bacteria. And this is because bacteria often produce natural products as a defense mechanism to help them kill off other bacteria, which they may be competing for resources in their ecosystem. And natural products can have lots of uses to treat cancer or as immunosuppressants, for example. But most of the time, they end up being antibiotics. So natural products make excellent antibiotic molecules for humans and have contributed a lot to the development of antibiotics through the years. But the problem with them is it's really hard to make variants by chemical synthesis. And this is because they're so extraordinarily complex. They're made by enzymes in the bacteria, and enzymes can do remarkably specific chemistry that's really hard to mimic for humans. So scientists have always been looking for other ways to try and get new antibiotics and new natural products out of these. And how do we do that in the modern era? Well, the most straightforward way is to find new bacteria to act as sources of natural products. So most bacteria in history have been found in soil samples, water samples, or infected human samples. But it's much harder to find anything from these environments these days, as a lot of them that ground has already been trodden. So scientists have gone to exotic places and extreme lengths to find new bacteria. Some recent examples include finding bacteria in Antarctica, volcanic environments, deep under the ocean, and other interesting marine environments, in search of new bacteria samples. When we find these new bacteria, what we can do is grow them in the lab in media. So media is just a solution of nutrients that allow the bacteria to grow. And then we can use techniques to find out what natural products these new bacteria make. Most commonly for this, we use liquid chromatography. So this allows us to separate out the mixture of compounds formed. Often there's a really complex mixture formed by the bacteria, so we need to separate those out. And we typically couple this with mass spectrometry, and this lets us find the exact mass of the molecules made, and hopefully the molecular composition. From there, we can find out what's interesting, see if there's any new things that we can get out of that, any new natural products. And from there, we can try and scale up the bacteria production, try and isolate those molecules and test them as antibiotics. Another method scientists have used is to look for what we call silent natural products. These are natural products where the bacteria have the set of genes necessary to produce the natural product somewhere encoded in their DNA. But when we actually grow them in laboratory conditions, these genes are switched off and we don't see anything being produced. And usually this is because the bacteria will only produce that product under a specific set of conditions, often as a response to something in its natural environment, something like having another bacteria that it's competing with nearby, or some sort of chemical signaling process. So what scientists have done is develop methods that can turn on these genes artificially. This can be done in a number of different ways. For instance, you can use specific organic molecules to switch on the genes and allow them to be expressed and the natural product to be produced. Or you can change the growth conditions of the bacteria. Changing the nutrient levels in the media can often turn on the genes. And a more advanced technique that has been used is to use state-of-the-art genetic editing techniques. So this allows you to change bits of the DNA sequence, and hopefully if you change them just specifically enough, you can actually turn the genes on and produce the new natural product. And once we've found a way to switch these genes on and activate production of the molecule of interest, we can use the same workflow of growing the bacteria in the lab and then use liquid chromatography coupled to mass spectrometry to analyze, find new molecules, and then test them for antibiotic activity. The final way to get new natural product variants, and this is the area I worked in, is to engineer the genes that the bacteria use to make the molecules. So you may know that if we have a set of genes, they will code for proteins. Typically, we represent proteins as shown on the right here. This is the so-called cartoon structure, where you can see the secondary and tertiary structure of the protein, with those sort of spirals being the alpha helixes in the secondary structure, and the arrows being the beta sheets. And proteins can have lots of different functions in different cells, but most of what we look at are enzymes that perform specific chemical reactions. And a set of enzymes will be encoded somewhere in the DNA of a bacteria that produces a natural product, and these enzymes will perform a specific set of chemical reactions that actually build up the natural product molecule. One of the things we can do as scientists is find these genes, make controlled edits known as mutations to them. This changes the set of proteins that are made in specific ways. And if we know what we're looking for and we target specific things, we can change the chemical reactions that the enzymes perform to make new molecules. So here's a real example with that of this particular molecule shown on the slide. One simple edit you can do is to change what specific enzymes are used. So in this case, the genes that install the methyl group in the original product have been swapped out for a set of genes that do the same chemistry, but with a starter molecule that doesn't have the methyl group in place, meaning that when you use this enzyme, the final product lacks this methyl group. That's a relatively simple change, so let's look at something a little bit more complex. So in this more complicated example, we've actually edited the genes to include a new enzyme activity rather than just change one. And here we've added activity from an enzyme known as a dehydratase, which has removed water from across the carbon-carbon single bond and turned it into a double bond that you might know as an alkene. And you can begin to see how interesting this can be, as this is just the engineering of one specific natural product at one specific position. And you can see how we could quickly explode this into lots of new natural product analogs if we look at more natural product gene clusters and we look at more editing to those gene clusters. And we're hoping to use this as a rapidly advancing way to make big libraries of natural product molecules that we can then test as antibiotics and find new drugs, discover new drugs. So let's quickly switch tacks and look at other ways we can make new antibiotics. And this is by looking at the man-made ways. And we typically refer to this as the medicinal chemistry, a more classical medicinal chemistry approach. And this is what I do now in my postdoctoral work here at the University of Oxford. So let's take a look at some of these molecules made in this way. You can see that they're often a lot smaller and less complex than natural products. This is due to humans, again, not having as good options for synthesizing molecules as bacteria do. But chemists can use clever targeting strategies to make very targeted edits to the molecules. And we can hopefully make these really good antibiotics despite their relative simplicity compared to natural products. So the big question you might have is, where do we even start with this approach? So with the natural products approach, we know that the starting point is a natural product that might already have some antibiotic activity. So where do we start when we don't know what sort of molecules we might be looking for? Well, let's look at the modern approach to this. So the first step to developing an antibiotic is knowing what you want to target. So how do we do that? Well, first we want to find some antibiotic-resistant bacteria. The easiest way to do this is to take swabs from infected patients or infected animals. We can then grow these swabbed bacteria in petri dishes filled with nutrient-rich media, which allows them to grow really well. We can then take that bacteria and we can use different methods to isolate its DNA. And from this DNA, we can then sequence it to get the genetic code of the bacteria itself. We can take the really long code that we get out of that and use computer programs to look for the specific genes that cause antibiotic resistance in those bacteria. And we usually do this by using the software to compare the genes to things that are already known about in the literature. So this lets us know what genes we want to look at. And once we found a gene that is particularly widespread and found in lots of different antibiotic resistant bacteria across the world, we can look at targeting that specific gene. And how do we do that? Well, the easiest way is to take that gene. We know that it encodes a protein, and we can actually produce that enzyme or a protein or enzyme in the lab. So here is an example of a protein gel from my lab. And on the left of the image, you can see a ladder of different proteins we use as molecular weight standards. On the right is a protein that I purified. So we can use this sort of technique to estimate size of the proteins that we have. We can also look at the purity of what we've produced. So here we get a nice single band. So this enzyme is really nice and pure and ready for us to test. So we can look at these enzymes in lots of different ways. We can look at the structure. So here's on the left of that is a structure that was taken of an antibiotic resistance enzyme. We can also look at the chemistry that those enzymes do. And ultimately, we want to look at that chemistry to help inform the development of molecules that block or inhibit the action of the enzyme. By removing the activity of that enzyme, we hope to remove the antibiotic resistance that it causes. And to do this, we develop molecules called inhibitors, which deactivate the enzyme, and we can look at how they deactivate the enzyme in the lab. So let's just talk about inhibitors for a second. So here's an example of an antibiotic resistance enzyme that we look at in my lab called beta-lactamases. They cause resistance to penicillin-like antibiotics that we mentioned earlier. And they do this by performing this chemical reaction, where they essentially perform a reaction similar to an ester hydrolysis. So they take an alcohol group, which attacks into this amide bond of the four-membered ring known as the beta-lactam. And this springs the ring open, hydrolyzes that ring, and this inactivates the penicillin as it can no longer act as an antibiotic because its structure has been changed. If we have an inhibitor with the penicillin, the inhibitor can actually stick to the active site of the enzyme and block it by binding really tightly to it. And now our penicillin molecule can no longer reach the active site of the enzyme. And in this way, the chemical reaction then can't take place, so the penicillin remains active and is able to be used in an antibiotic as its structure remains the same. And an example of this on the market is actually this molecule on the right here called avibactam. This is a molecule that it does exactly this. It inhibits beta-lactamases. And it's often co-administered to patients with penicillin-like antibiotics. So by doing that, you have the avibactam and the penicillin administered to the patient. When it gets to the bacteria, the avibactam protects the penicillin from this beta-lactamase resistance mechanism and allows the penicillin to kill the bacteria, even if it is resistant, normally resistant. So the final step of the process is we take the inhibitor molecules that we find, we can optimize them to hopefully produce new medicines. So now I put a question mark next to new medicines because it isn't just as simple as finding molecules that are good at inhibiting enzymes or stopping the bacteria from being resistant. There are countless factors to consider before something is made into a medicine. The main ones being, is it toxic to humans? Does it produce really nasty side effects that will make people not want to take the medicine? And also, does the molecule actually get into the body? And specifically for antibiotics, does it get to where the infection site of the bacteria is? So if the bacteria is infecting a specific part of the body, can we actually deliver it there? Does the molecule get there in the first place? And these are problems that need to be tested for in the lab, but they can be solved by going back to the molecules that we produce, updating the molecular structure by synthetic chemistry, And hopefully, we can develop something that is both a good inhibitor for these antibiotic resistance enzymes, but also a good medicine for use in humans. So, the part of the process I work in is this bit highlighted here. So, I take enzymes that cause antibiotic resistance and try and work out ways that we can test them and hopefully find new inhibitors. So, how do we do this? To look at and find enzyme inhibitors, we need what is called an assay. An assay is just a fancy word for a test or an experiment on that enzyme, usually with some sort of quantitative output, so some numerical output that we can measure. And we can do this in two ways. The first is to use an activity-based assay, and this is just a way of saying that we look at the chemical reaction that the enzyme actually does, and we can monitor that in some way or another. Usually, this is done by looking at the conversion of the starting material of the enzyme to its product. So if we add an inhibitor, then we know that less product will be formed, and that activity we can say has been reduced. And the less inhibitor you need to stop the product formation, the better and more potent that inhibitor is, because you need less of it to elicit the same response. So activity-based assays are typically the most common way to look at enzymes, as they're usually very representative of what the enzyme does, but they're also very easy to understand. Oops. But sometimes activity assays can be complicated for a number of reasons, so we need other ways to look at them, other ways to look at the enzymes. The second most common option is what we call a binding-based assay. What this means is rather than look at the chemistry that the enzyme performs, we can actually look directly at how the inhibitors stick to the active site of the enzyme. So this is usually a good approximation for the activity, as you can assume that a molecule that sticks really tightly to the active site is probably going to stop the chemistry that it does from happening, and therefore it's probably going to act as an inhibitor. And this is the method that I have been using to study and identify enzyme inhibitors. So the technique I use to study the particular enzyme I work on involves a concept called fluorescence, which you may have met in chemistry or physics classes. So if we take a molecule like fluorescein here, you can see it has a lot of benzene rings connected together. What this does is create a lot of delocalized electrons around those rings that cover a large amount of atoms. And this property of the molecule allows it to absorb high energy, short wavelength UV light. When it absorbs the energy from the light, it's transferred to the electrons. Some of that will dissipate out as heat, as transferred as heat energy, but some of that energy remains in those electrons. And what happens is that when you shine a light onto a fluorescent molecule, you get an emission of a light of a longer wavelength. So for instance, in the case of fluorescein, when you shine blue light at about 480 nanometers, it will emit longer wavelength, lower energy light at around 525. And you get this blue excitation light leading to a green emission. And you can plot this as a graph. So here is the blue line representing the wavelength of light absorbed by fluorescein, known as the excitation. And the red line is the wavelength of light that gets emitted by the molecule once it has been excited by the light, represented by the blue line. So one of the properties of light you may know is that it acts as a wave, and usually that wave can oscillate in multiple directions known as planes. If we pass that regular light through a special filter, we can actually only let light in one of those planes be produced. We call this plane polarized light, and this is where the light is only oscillating in one of those planes. You might know these from your sunglasses. They often have things called polarized filters, and this is essentially how they work to block the light from getting to your eyes. Interestingly, if we shine plane polarized light onto a fluorescent molecule dissolved in a liquid, it gets absorbed as normal and emitted, but the emission actually loses this polarity, meaning it goes back to oscillating in one plane to multiple planes. And this is due to the molecule rotating in solution. And this rotation causes the scrambling of that polarization. And we often call the emission depolarized because it's lost that polarization. And the degree to which this happens depends on how fast the molecule rotates in solution. And how fast the molecule rotates in solution is directly proportional to how big it is. So small molecules like fluorescein rotate really fast and depolarize the emission a lot. But a bigger fluorescent molecule might rotate slower, and that will depolarize the emission less. And we can use this phenomenon to exploit and study. We can exploit this phenomenon to study enzymes. So in my work, we found an antibiotic resistance enzyme we wanted to target. We knew what the substrate of the enzyme was, so we attached a fluorescent group, such as the fluorescein, to it by chemical synthesis. And we know that as a smallish molecule, this fluorescently tagged substrate rotates really quickly in solution, and therefore you get this depolarized emission. However, when we add the enzyme, we know that the substrate binds really tightly to the enzyme active site, and this complex of the enzyme and substrate is actually really, really big. We know that enzymes are massive macromolecules that range from thousands to tens of thousands to hundreds of thousands of Daltons. So enzymes rotate very slowly in solution compared to small molecules. So when our substrate is tightly bound to the enzyme, it rotates a lot slower too. So more of that fluorescent emission remains in the plain polarized state. So it gets less depolarized. And we can actually measure the difference between those two states. So here's some data from my work. So along the x-axis is the concentration of enzyme with a fixed concentration of our fluorescent substrate. And on the y-axis is the polarization, with a high value being polarized and a low value being depolarized. So we see at low concentrations of enzyme, we get very little polarization because we've got this fast rotation leading to depolarized emission. But as we add more and more enzyme, our fluorescently tagged substrate binds the enzyme and it rotates a lot slower, leading to more polarization. And as we increase the enzyme concentration, that polarization increases until it reaches a specific point where it starts to plateau. And this is because all of the fluorescent substrate is already bound to enzyme, so adding more enzyme doesn't actually help things, doesn't really change things anymore. And we can use this effect to study inhibitors. So if we have a good inhibitor, it's going to stick to the active site of the enzyme, and this pushes our fluorescent substrate into solution. So when it's in solution, it can rotate much faster than it does bound to the enzyme, and this depolarizes the emission of the fluorescence. On the other hand, if we have a bad inhibitor, something that doesn't stick to the active site very well, the fluorescent substrate stays complexed with the enzyme, rotates slower, and keeps the polarized emission. And again, we can measure this difference. So on the right here is some data for a good inhibitor from my lab. And you can see along the x-axis, we've got concentration of inhibitor this time. And on the y-axis, again, we've got change in polarization. So this is high polarization is a polarized emission, and low is depolarized. So you can see that as we add more and more of this particular compound, the polarization decreases as the inhibitor binds to the enzyme, pushes that substrate into solution, and this depolarizes the signal that we get. And typically what we measure from this is something called the inhibitory concentration at 50%, otherwise known as the IC50. And this is the concentration of inhibitor that gives you 50% response in your particular assay. And we can compare IC50 values between different molecules, and this will help us find the best inhibitor. So a low IC50 is usually better. Well, it's always better actually, because having a lower IC50 means you need less of the inhibitor to get the same response. So less inhibitor to get the same amount of inhibition, essentially. So in my lab, we're using this exact approach to do what's called a high throughput screen. And this is just a fancy way of saying testing lots of molecules as quickly as possible and seeing if they're good inhibitors. So we're taking a similar setup like this. So finding our enzyme that causes antibiotic resistance with a fluorescently tagged substrate, and we're mixing with lots of different compounds to see what gives us good inhibition. And to do this, we've had to miniaturize the assay, and we've adapted it for use in these things called microplates. So there's a diagram here, and these are about the size of 2 credit cards stuck together, and they have space for 384 simultaneous experiments. So you can imagine that we can get through a huge amount of experiments really, really quickly. And we've also miniaturized it to just 25 microliter reactions. That's about a drop of liquid in each of these different wells of the microplate. And we can test more than 300 compounds in each of these plates. And hopefully we are going to start screening molecules to see if we can get inhibitors, and we're going to try and do over 100,000 different compounds in the coming months. There's a huge amount of molecules. The reason we test so many molecules is because we know that enzymes have such a specific active site structure that only very few molecules will actually stick to it. So we'll have a very low hit rate, which is why we need to screen lots of molecules to give us the best chance of finding new inhibitors. And hopefully for our target, we'll find some new inhibitors, or at least some new starting points that we can develop into better inhibitors along the way. And we can do this using lots of different chemical synthetic methods. So that's all that I would like to talk to you about in terms of science. So you may be wondering, I trained as a chemist, but a lot of the work that I do is now very biological and very biochemistry leaning. So was it worth me doing a chemistry degree? Why didn't I? Should I have done a biology or a biochemistry degree instead? Well, I disagree, because I think my training in chemistry really helps me on a daily basis to be a better biochemist and understand my biochemistry work. For instance, enzymes are essentially really big organic molecules that perform organic chemistry reactions. So understanding organic chemistry really well helps me to understand what the enzymes are doing to a degree I think someone with a more biologically relevant training might not appreciate. And it also helps you to design new antibiotic molecules. So we have teams here at the University of Oxford who develop and synthesize new antibiotics, and having a training in organic chemistry means I kind of speak the same language as them, and we can work together really efficiently. Also, during my degree, I learned to do a lot of analytical techniques such as mass spectrometry and NMR spectroscopy. And I still use these to this day to help me understand complex chemistry that enzymes do, as well as to design assays against enzymes that use these similar analytical techniques. And we still use these analytical techniques to really push forward our work. And finally, all the problem solving and mathematical skills that we mentioned are skills that a chemistry degree taught me, and they still really help me out on a day-to-day basis. So for instance, designing assays requires a lot of statistics and a lot of data processing skills. So maths for me really plays a huge role there and in many other places, and I may not have got that without training in chemistry. So all that remains for me to do is to thank the 2 labs where I've done a lot of my research. So first off, Greg Chellis and Professor Yusuf Lewandowski at the University of Warwick, where I did my PhD. And now in the lab of Professor Chris Schofield here in the INEOS Oxford Institute at the University of Oxford, and all the people that I've worked with along the way. And also to thank you for your attention, and I believe there will be a question and answer session following this video shortly. So thank you for your attention, and goodbye.